Hello everyone, this is Katish here. In this video, I'm going to go through the AQA Biology Paper 1 Higher Tier, question number 6, which is on the topic of bioenergetics. This question is about photosynthesis. Complete the symbol equation for photosynthesis. So they've already given you a balanced equation. So they're asking merely for the reactants and one of the products. And it's a symbol equation. Remember, not the word equation. So you're not going to get any marks if you write the word equation. So the reactants for photosynthesis would be carbon dioxide, CO2 plus water, H2O. Now, doesn't matter which order you write that in. And this is glucose and 6 oxygen. Right, so that's glucose. Figure 6 shows how the rate of photosynthesis changes with light intensity. So you've got a line graph with the rate of photosynthesis in arbitrary units on the y-axis and the light intensity in thousands of lux. So when it says 5, so it's 5,000 and 10 is 10,000. Now let's have a look at the graph. The straight line somewhere up to about 7.5. So if I attempt to draw a straight line, does it become straight? No. Let me redraw it so you can see that it's actually a straight line up to about, there you go, up to about 7. And then, so up to about this point, it's actually a straight line. And then it slowly, the rate is actually, at this point, the rate is constant increase. And the increase in rate is actually slowly decreasing. And after this point, the rate is the same or constant. Constant rate. Right? This is constant increase. Right? Here it's rate increase is decreasing. Right? The rate of photosynthesis, how it is increasing, is actually getting smaller, lower and lower. Right. So which part of the graph could be represented by the equation y equals mx plus c? Now this is the first time actually seen a link to a mathematical equation here. That is, this equation is representing a straight line. Right. So where is the point where it's actually increasing? So it's basically between 0 and about 7. So the options that you have is 0 to 5,000, which would be the answer. A student investigated the effect of color on light on the rate of photosynthesis in leaves. Figure 9 shows how the investigation was set up. So this is a common practical that is usually done when investigating photosynthesis. So you have a lamp and you have sodium hydrogen carbonate solution, so that you have the hydrogen carbonate provides the CO2 for photosynthesis. It's already in solution, so you've got the water for photosynthesis. So you've got those two. The disc cut from a leaf, so you're actually cutting similar sized parts of a leaf, right? And the results, table 5 shows the results. Color of light blue, time taken for 10 leaf discs to reach the surface of the solution in seconds. Now, what does this actually mean? So here, when these leaves are photosynthesizing, it is going to produce oxygen. Now, those oxygen might not actually be given out. So you might not actually see bubbles of oxygen. They might simply stick to the surface of the leaf. when it sticks to the surface of the leaf, then the leaf would actually become buoyant and it would actually float to the top, right? And then once the oxygen bubbles burst, it might actually sink down. So the student is waiting for 10 of these discs to float to the surface and timing how long it actually takes. So blue is 115 seconds, green is 831 seconds, red is 397 seconds. So the shortest time is actually blue, which means most photosynthesis is taking place at blue, and the least is in green light. Now let's have a look at the question. Give one way the student could change the color of light shining on the leaf disc. Now you could use different colored light bulbs. So here you got a lamp. You could change the light bulb, or you could use place a filter in front of the lamp or in between. So let's say you want a green filter, green color filter. So you would place a green filter in front of the lamp, so it actually shines green light. So there's a couple of different answers. Here's all the different, right? Use different colored light bulbs or LEDs. LEDs, it's much easier to change the color. Use different colored filters in front of the lamp, or put different colored transparent materials over the lamp and the beaker, which is basically the same as saying put different filters in front of the lamp. Next question. Give the independent variable and the dependent variable in this investigation. Now the independent variable is what you're changing, so that would be the color of light. The dependent variable is what you're actually measuring, which is the time taken for 10 leaf discs to rise to the surface of the solution. All of the air had to be removed from the leaf disc before placing them in the beaker. Suggest one reason why. Now, if you don't remove the air from the leaf disc before placing it in the beaker, then they will simply float. Right? So you want the leaf disc to be at the bottom of the beaker before you start the investigation. So here is a typical answer. So the disc would sink to the bottom of the beaker. That would give you the mark. Or you could say, so any gas that makes the disc rise to the rise is from photosynthesis, or air is a gas, so any left in disc would add to the oxygen produced by photosynthesis. I think the first option would be the best. The leaf discs were placed in a beaker of sodium hydrogen carbonate. Explain why sodium hydrogen carbonate solution was used instead of water. Sodium hydrogen carbonate would break down to produce the carbon dioxide, and that means it's not going to be a limiting factor during this investigation. Explain why the leaf disc moved to the surface of the solution during the investigation. Now, this is one of the reasons they didn't explain why or the method clearly at the start. So, as I explained at the start, oxygen was produced in photosynthesis. The oxygen, or the gas entrapped in, is trapped in or around the leaf disc, which causes the leaf disc to move to the surface. Right, let's have a look at the next part. There are two types of chlorophyll in leaves. Figure 10 shows the percentage of different wavelengths of light that the two types of chlorophyll absorb. So the percentage of light absorbed is peaking at this point. The dotted line is chlorophyll A. So that's A, and this is B, and speaking over here. So this, so 400 to 500, so each box is 10, so that's 440, and this is speaking at 490, and let's have this one, that's 640, and that's 675. Okay. Table 6 shows the color of light waves. Sorry. Table 6 shows the color of different wavelengths of light. So anything between 380 to 435 is violet. So 435 is going to be violet. And 450 to 499. So if let's see. So 380 to 435. So that's 435 is going to be up to this point. That's 435. 450. So this is violet. 450 to 500 is going to be blue. 500 to 570, so here is going to be green. 571 to 590, that's going to be yellow. And 590, it's slightly going to be on this side, and 620 to 720 is going to be red. Okay, now it's important that you spend a little bit of time to interpret the graph the best way you can before you start trying to answer the question. So suggest the advantage of a plant of having two types of chlorophyll. Now if it has only one type of chlorophyll, that means it's limited to the amount of photosynthesis it can. If it has two different types of chlorophyll, that means it can absorb two different wavelengths of light, and the wavelengths of light changes during the day, from the time the sun rises to the sun sets. So it is trying to maximize the amount of photosynthesis a plant can actually do. So having two types of chlorophyll will maximize it. So to absorb, you use many colors of wavelengths of light rather than just one wavelength of light. Table 5 is repeated below. So you've got the same table. The leaf discs in the investigations are green color. So explain the results in table 5 for blue light and for green light. Use the data from table 10 and table 6. So figure 10 and table 6. Figure 10 and table 6 is basically the graph and the table here. So let's go back to the question. Explain the results for blue light and for green light. So as you can see, blue light is 450 to 499, and green light is from 500 to 570. So now blue light, chlorophyll B has a maximum photosynthesis. Percentage of light is absorbed by chlorophyll B. Chlorophyll A is still managing to absorb about 30 to 35% chlorophyll A, and then but it steadily drops by the time chlorophyll B actually peaks. Now green light, both of them, the percentage of light absorbed is 0 because it's a flat line at this point. So you can see, so it's a flat line at this point for green light. So blue light is actually peaking at this point. While at the start of the blue light, the percentage of light absorbed by chlorophyll A is actually reducing, whereas chlorophyll B is actually peaking at these points, right? So let's go back to the question. The leaf discs in the investigation are green, which explains this. The time it took for the 10 leaf discs to rise to the bottom is 831 seconds, whereas blue light, it was the lowest. So the rate of photosynthesis was highest here because the percentage of light absorbed by chlorophyll B is higher during blue light. So chlorophyll B is actually working maximum here. Okay. Explain the results. Here's a standard answer. Chlorophyll absorbs most or a lot of blue light. Chlorophyll absorbs least or very little or not much of the green light. So discs in blue light took the least time to rise to the surface because they are photosynthesized faster or more. So discs in green light took the most time to rise to the surface because they photosynthesized slower. So you have to use data from figure 10 and table 6. Example, approximately 80% of blue light was absorbed. So you'll have to refer to the data as well here. So I think, let me move these points, which makes a bit more clear. Chlorophyll absorbs most or a lot of blue light. So discs in blue light took the least time to rise to the surface because they photosynthesize faster or more. Chlorophyll absorbs least or very little or not much green light. So discs in green light took the most time to surface because they photosynthesize slower. And also mention the data from the table and from figure 10 as well. That brings us to the end of this question and to the end of the paper. Thanks for watching. I'll see you in the next video.